Right.
Okay, so excellent. All right, so these are the lipids. So you see the acyl chains, the hydroxy fatty acids in the human body. What I'm going to tell you about today is the discovery of this class of lipids, their biologic actions, beneficial effects in type 1 and type 2 diabetes, and the regulation of the tissue levels aimed at developing them, hopefully, as a treatment for metabolic or immune-mediated diseases. Now, the key actions are they augment glucose-stimulated insulin secretion. They increase insulin-stimulated glucose transport into fat cells, and they are anti-inflammatory. So my lab has been interested for many years in what links obesity to type 2 diabetes. And as we know, there are many factors involved in obesity and type 2 diabetes. There are genetic and epigenetic factors. Diet plays a role, lifestyle, exercise, environment, circadian clocks, the gut microbiota, more and more evidence it's important. All of these affect metabolism, and metabolism affects adipose tissue, but adipose tissue also affects metabolism. So changes in fat cell function alter insulin sensitivity in the liver and muscle, insulin secretion from pancreatic beta cells, and food intake and energy expenditure through the brain and the sympathetic nervous system. So we started our story wondering about glucose transport in adipose tissue. And we were interested in GLUT4, the major insulin-regulated glucose transporter, primarily expressed in adipose tissue and muscle. It is possible to find it under some conditions in other tissues. But what we and others observed in humans and rodents in obesity and type 2 diabetes is that GLUT4 expression goes down in adipose tissue, but not in muscle. And this seemed really puzzling at the time. Could this change in GLUT4 in adipose tissue contribute to diabetes, since very little glucose goes into adipose tissue? But we were interested because of data like this. This was a collaboration with Ulf Smith in Sweden, and we showed that the GLUT4 levels in the adipose tissue correlated highly with the insulin sensitivity measured by euglycemic clamp in people. And Ulf went on to show that decreased GLUT4 protein levels in adipose tissue are an early predictor of type 2 diabetes in people. And recently, in 2023, there was this very interesting GWAS study where, with rigor, they had 55,000 participants, 3 different ancestry groups, and different from previous studies, which looked at insulin resistance in the fasting state, during which hepatic insulin action predominates. This study focused on insulin levels measured 2 hours after a glucose challenge, when insulin action in muscle and adipocytes predominates. And what they found was 9 new candidate genes for type 2 diabetes in humans that regulate the expression or trafficking of the GLUT4 glucose transporter. Trafficking, we mean from intracellular vesicles to the plasma membrane. So these studies, these genetic results tell us that GLUT4 is important for insulin sensitivity and type 2 diabetes risk in people. But does GLUT4 in adipose tissue actually affect systemic insulin sensitivity? And to address that, we made mice with adipose-specific knockout of GLUT4 or adipose GLUT4 overexpression. The GLUT4 expression was still normal in the muscle of these mice. And surprising, when we did this years ago was that adipose-specific GLUT4 knockout caused insulin resistance with increased risk of type 2 diabetes, whereas adipose-specific GLUT4 overexpression was associated with enhanced glucose tolerance and lower fasting glycemia. And when we studied the tissues in vivo and ex vivo, there was evidence for systemic effects on muscle and liver from this defined genetic alteration in fat. So we did a series of studies, including DNA array analysis, which was the untargeted way to approach this in those days. And what we found was when you increase GLUT4 in adipose tissue, that is sensed by this transcription factor, carbohydrate response element binding protein. It's a transcription factor that regulates lipogenesis and glycolysis. That results in more glucose being driven into fatty acid synthesis or de novo lipogenesis. And that has favorable effects on insulin action on the liver to suppress hepatic glucose production and insulin action to promote glucose utilization in muscle. And we know ChRABP is key because when we knock it out only in the fat, we no longer have the increase in de novo lipogenesis, and we no longer have the favorable effects on liver and muscle. So we asked, what are these fatty acids with beneficial metabolic effects? And we teamed up with Alan Sagatilian, who was in the chemistry department at Harvard, and his student, Edward Holman. And we did untargeted lipidomic analysis. And we were looking for new lipids. And what we found turned out to be branched fatty acid esters of hydroxy fatty acids. So we call them FAFAs. So let me show you, they consist of an acid fatty acid group, here palmitic acid, and a hydroxy fatty acid. Here I'm showing hydroxysteric acid bound by an ester bond. So this is 9-palmitic acid hydroxysteric acid, or 9-PASA. And actually, this ester bond can be at a number of different carbons. So there are multiple PASA isomers with branching at different carbons. Now, we don't see it at the alpha or the omega carbons, but we see it at the internal carbons. So now we know there's actually a whole FAFA-ome. And a colleague, Andrzej Kuda, at the Czech Academy of Sciences in Prague, made this diagram. And the FAFAs are organized around the hydroxy fatty acid. And you'll see it can include arachidonic acid. It can include polyunsaturated fatty acids. There are many different types of fatty acids that can be incorporated into FAFAs. So there are also additional FAFA families in plants, and FAFAs are evolutionarily conserved and are present even in yeast. So we have studied mainly the palmitic acid hydroxysteric acids. So what is the tissue distribution of the PASA family of lipids? They're in every tissue we've looked at, with the highest levels in white fat and brown fat, but present in liver, pancreas, kidney, brain, skeletal muscle, heart. Tissue concentrations are similar to other signaling lipids like prostaglandins and prostacyclins. So our PASA is regulated in insulin-resistant people, and we collaborated again with Ulf Smith in Gothenburg, and he sent us serum from insulin-sensitive and insulin-resistant people. And you can see the FAFA isomers are reduced in serum of insulin-resistant people. And when we compared it to insulin sensitivity, again, measured by euglycemic clamp, we saw some incredible tight correlations on the order of 0.9 for 1312 PASA and 5 PASA, correlation between serum levels and insulin sensitivity in people. So I asked all for fat biopsies from these people, and you can see the PASAs are also reduced in the adipose tissue, and the adipose levels also correlate with insulin sensitivity, but the correlations are not quite as strong for adipose tissue as they are for serum, but still 0.9, 0.798, 0.71. So other people have looked in other human settings. So the Kuda and Kopecki in Prague found that levels of palmitic acid, hydroxystearic acid are reduced in breast milk of obese mothers. Another group looked at exercise training and showed that PASAs actually go up in adipose tissue, also in serum, when elderly women are put on a training regimen. And there's this other interesting paper with acute aerobic exercise in overweight and normal weight runners. And what they find is many FAFAs, many families change, and the profile is different among the normal weight and the overweight participants, but the FAFAs could predict VO2 max, and they could modulate inflammatory responses, fuel utilization, and insulin resistance. So what are the biologic effects of PASAs? Well, you know, many lipids play a structural role. Other lipids are used for energy via beta oxidation. And then there are signaling lipids. I mentioned the prostacyclins. And we think that FAFAs are signaling lipids. So what are their biologic effects? Well, as I mentioned in the beginning, they increase glucose transport in fat cells. They decrease inflammation, both directly on macrophages and dendritic cells, but also some effects on T cells. They decrease hepatic glucose production. They increase GLP-1 secretion from gut enteroendocrine cells. They also increase glucose-stimulated insulin secretion from islets. And then there's the secondary effect through the GLP-1 on insulin secretion. And most recently, we've shown that they have direct protective effects to decrease senescence in pancreatic beta cells. So all of this then improves glucose homeostasis. Now, I want to be really clear that the secretory effects, GLP-1 and insulin, we see in insulin-resistant aged chow-fed mice, and we see in human islets, but we don't see this in mice on a high-fat diet. Instead, in mice on a high-fat diet, the beneficial effects of PASAs result mainly from their insulin sensitizing effects. And I think this is important because insulin resistance itself is a major risk factor for cardiovascular disease, cognitive function, and cancer. And other treatments for diabetes are important, but there are people with insulin resistance who don't have diabetes who might benefit from reducing insulin resistance. So what is the evidence that PASAs are insulin sensitizers? So for this study, Anna, Pong, and Odile chronically PASA-treated high-fat diet mice for 13 weeks. There was no difference in weight gain or adiposity. And then they performed the euglycemic hyperinsulinemic clamp to measure insulin sensitivity in vivo. So here is the glucose infusion rate. And you can see in vehicle-treated high-fat diet mice, very little glucose needs to be infused to maintain glucose levels with insulin infusion. But when mice have chronically been treated with PASAs, their insulin sensitivity is markedly improved. And if we focus specifically on the liver and the ability to suppress hepatic glucose production, in vehicle-treated mice, hepatic glucose production is only suppressed about 8%. But with PASA treatment, we can markedly enhance the suppression of hepatic glucose production. So PASAs enhance hepatic and systemic insulin sensitivity. Now, they also have modest effects to increase glucose uptake in skeletal muscle and in the heart. So PASAs improve insulin sensitivity in multiple tissues. How can they have so many effects? And the answer is that they work through lipid sensing G protein coupled receptors. And there's growing data for how GPCRs have multiple roles in glucose homeostasis and diabetes. So GPR120 is in neuroendocrine cells. It's involved in GLP-1 secretion. It's also in macrophages and it has anti-inflammatory effects. It's also in adipocytes and alters adipocyte differentiation and glucose uptake. And other GPCRs, GPR 40, 43, 119 are in pancreatic beta cells and modulate insulin secretion. So we showed that PASAs activate GPR120, which mediates the effects on glucose transport in fat cells. They also activate GPR40, which mediates the effects on insulin secretion. And there's a G-alpha I receptor that mediates the PASA effects on hepatic glucose production. So PASAs have quite striking anti-inflammatory effects. And we first studied this in the setting of ulcerative colitis, because when Jen Lee was in the lab, she was very interested in the gut. And she and Pedro Vieira showed that PASA treatment delays the onset and reduces the severity of colitis. And it markedly reduces gut inflammation. So normally in colitis, dendritic cells present antigen to CD4 T cells, which stimulates IL-12 release, which results in CD4 T cell activation and clonal expansion, and the release of interferon gamma, which drives gut inflammation. So what Pedro showed is that PASAs reduce co-stimulatory molecule expression and dendritic cell activation. So there isn't as much clonal expansion. There isn't as much interferon gamma. And Jen showed this results in less gut inflammation. So could these anti-inflammatory effects be beneficial for other immune-mediated diseases? What about type 1 diabetes? And Ismail Saeed in the lab studied type 1 diabetes in the NOD mouse. So in our colony, 83% of the female mice will get diabetes by around 26 weeks of age. But if we treat with PASAs, that delays the onset and markedly reduces the incidence of type 1 diabetes. It also improves survival in these mice. And what we showed is that there are two categories of mechanisms. There's immune cell modulation, and there are also direct protective effects on pancreatic islets. So here is an islet from an NOD mouse treated with PASAs. And it's a pretty normal looking islet with the magenta insulin staining cells filling most of the islet and these green glucagon cells around the periphery. This islet is not being attacked by immune cells. Another islet in the same mouse is under immune attack. But here's an islet from a vehicle-treated NOD mouse. So it's very elongated, and the glucagon staining cells are intermingled with the insulin staining cells, probably because of the loss of insulin-producing cells. And this islet is under quite extensive attack. We looked at beta cell proliferation in these islets using Ki-67, and you can see PASAs increase beta cell proliferation. And we wanted to know if these effects were really direct effects or if these were effects that depended on the immune system. So we then carried out studies in Min6 clonal beta cells where there is no immune system, and we stimulated them with cytokines, which increased apoptosis, and then treated with PASAs, which reduced that to a large extent. The same was true with necrosis, increased with cytokines, decreased with PASAs. And when we looked at beta cell proliferation, that was decreased with cytokines and partially restored with PASAs. So we think that there really are direct effects on the beta cell. So beta cells are prone to endoplasmic reticulum stress, and this is a factor in both type 1 and type 2 diabetes, and also a stimulus for cellular senescence. So Maria Fernandez stained these pancreas sections for XBP, which is a marker of ER stress. So you can see the red staining in the vehicle-treated decreased with PASA treatment. Here's the insulin staining. You can hardly find it in the vehicle-treated NOD mouse, but lots of insulin staining with PASA treatment. And when you do the merge, you can see that there's reduced XBP in insulin staining cells. Here is the quantitation, and she also looked at CHOP, another marker of ER stress. So PASAs reduce ER stress in islets in vivo. Now, what about in human islets? And for this experiment, Ismail Saeed induced ER stress in human islets with thapsigargan, a chemical agent. And then he showed that 9 PASA partially prevents and 5 PASA prevents even more the ER stress in human islets. And this has functional consequences. So in these islets, you can see here's normal glucose-stimulated insulin secretion. That is lost with cytokine treatment, but it is at least partially restored with PASA treatment. So we really wanted to know what's going on here. How do PASAs protect pancreatic islets under metabolic stress? And the stresses could be endoplasmic reticulum, glucose toxicity, cytokine lipotoxicity. All of that is present in the NOD mouse. And so we decided to do bulk RNA sequencing in the islets from the NOD mice treated with vehicle or PASAs. And we specifically designed this to start the treatment at 5 weeks and end it at 10 weeks before the mice had become diabetic, because we didn't want to confound the findings with hyperglycemia. We also sorted out the immune cells from the islets. So what we were looking at was primarily endocrine cells. So here are 5 different vehicle-treated mice. Lots of genes are lowly expressed at low levels, many genes at high levels. Now, 4 out of 5 of these mice are going to get type 1 diabetes. So how do PASAs prevent that at the gene expression level? And you can see marked alteration in gene expression for hundreds of genes with PASAs. So what are these genes? Well, some of them are islet genes. Here's the insulin 2 gene. Here's urocortin 3, a key islet marker. Here's glucagon. So it's not all beta cells, but also, alpha cells. And then there are the so-called disallowed genes, those that are usually not expressed. And you can see PASA's lower expression, LDHA, is one example of a disallowed gene. In addition, we found that many genes involved in senescence were decreased by PASAs. And there's something called the senescence-associated secretory pathway, or SAPS. SAPS is a production of a complex mixture of secretive factors that include pro-inflammatory cytokines, chemokines, growth factors, and proteases. And these are also reduced with PASA treatment. So senescence is involved in beta cell failure and destruction in both type 1 and type 2 diabetes. So we thought this was very interesting. We went to the literature and we educated ourselves a little bit about this. So in general, in healthy beta cells, you see beta cell markers and protective genes, and these disallowed genes are not expressed at all. But then with metabolic stress, beta cells start to undergo senescence, you see a decrease in the beta cell markers, and you see an increase in the disallowed genes, the senescence genes. Beta-gal is a marker for this. There's an upregulation of anti-apoptotic pathways, and then the expression of the SAPs. And the SAPs then feeds back on the not yet affected cells to damage those cells. So senescence is a hallmark of aging, but it differs because the cells do not die. They lose functional maturity and they release the SAPs, which then damages other cells. So I just wanted a little bit of sort of clinical flavor here. Senescence has actually been shown to be involved in many diseases affecting different organ systems. Senescence markers have been measured in humans with these diseases. These knockouts of key senescence mediators in mice have been shown to change the course of these diseases. And there are senolytic treatments now being used in animals, but also in people to treat some of these diseases. So this is a promising pharmacologic area, I think. And some people are actually targeting senescence in adipose cells. So senolytic agents kill senescent cells, and they abort the SAPs, and they limit the related tissue destruction, but they also reduce cell mass. So you can imagine for type 1 diabetes, that's not great, reducing the mass of beta cells. And for neurologic disease, it's not that advantageous. So we're looking for xenomorphic agents that don't kill the cells, but reverse them to functional cells. So how do PASAs protect against senescence? Well, DNA damage is upstream of senescence, as are reactive metabolites like ROS, ceramides, fatty acids, and high glucose. And these can activate either the P16 pathway or the P53 pathway resulting in senescence. And I wanted to point out that DNA damage is both upstream and downstream of these pathways. So which pathway do PASAs affect to reduce senescence? And I will say it's the P53 pathway. So what's been shown is that metabolic stressors like cytokines induce P53, and that induces senescence. And P21 is a key readout, and IL-6 is a readout for the SAPs. So what we found in our DNA array analysis was that the 5 and 9 PASA treatment in the NOD mouse increased MDM2, a negative regulator of P53. So that then reduces P53 expression and reduces senescence. So how did we prove that it was this MDM2 pathway? Well, we used siRNA. So here are islets in a dish, and you can see cytokines induce this marker of senescence, P21. They also induce this marker of SAPs, IL-6, and PASA treatment prevents that induction for both the P21 and the IL-6. Now, what happens if we treat with siRNA to MDM2? We no longer have the protective effects of the PASA on P21 or IL-6. So PASAs reverse senescence by enhancing expression of the negative regulator of P53, MDM2. Is this relevant to human islets under metabolic stress? So in this experiment, Maria had 6 different donors, normal donors of islets. Each one has a different symbol. And she treated those human islets in culture with cytokines. So you can see that induced senescence in all of the islets. And then she treated with PASAs. So this was actually a reversal experiment. not even prevention. And you can see that even the lowest one goes down with PASA treatment, and the red one goes down, and the black one. All of the people decrease their senescence markers with PASA treatment. And here are the SAPs. Again, human islets, PASAs, prevent and reverse senescence. So PASA treatment reverses cytokine-induced senescence in human islets. So for this part of the work, we would say that in mice and in humans, metabolic stress induces senescence in islets, but PASA treatment results, and I didn't show you these data, in increased DNA repair, increased glutathione, which has an antioxidant effect, decreased senescence, and decreased secretory pathway from senescence. So the PASAs reduce beta cell senescence and preserve beta cell phenotype by decreasing DNA damage. And they also preserve beta cell mass, so they are xenomorphics, not senolytics. All right, so how are the FAFAs and the PASAs regulated in mammalian tissues? And again, FAFA is the name for the whole class of lipids, like cholesterol is the name for all the cholesterols. PASAs are one family. So we got very surprising results with fasting and refeeding. So these are different PASA isomers in adipose tissue. And you can see most of them go up with fasting. This is really unusual, right? Fatty acids go down with fasting because lipolysis stimulates release of fatty acids from triglycerides and they leave the cell. But FAFAs are going up. And it's somewhat isoform specific. We don't see it with these two isoforms. These levels are restored to normal with 4 hours of refeeding, and this is tissue specific. We don't see the increase in the same PASAs in the liver. So how are FAFA levels regulated so rapidly in altered metabolic states? And we think that synthesis and degradation are part of it. But Dan Tan, a postdoc in the lab, was very intrigued with the idea of whether they're incorporated into other lipid classes that could work as storage depots. And in particular, she wanted to know if FAFAs got incorporated into triglycerides. So she did an experiment to see if there were FAFA-containing triglycerides in fat. So the idea is that triglyceride is made up of a glycerol backbone with 3 fatty acids. And what Dan actually showed is one of these acyl chains could be a FAFA. Rather than a simple fatty acid, it could be a fatty acid hydroxy fatty acid. And then she showed that FAFA-containing triglycerides can be synthesized in adipocytes. And I'm showing Dio as well because he synthesized the standards to be able to do these mass spec experiments. So triglycerides release fatty acids when lipolysis is stimulated, such as with fasting. So can lipolysis regulate FAFA levels? So this is a lipolysis experiment in cultured adipocytes. We stimulate lipolysis with forskolin or IBMX, and we add inhibitors of lipolysis. So here's the stimulation, and here's the marked reduction in lipolysis with an inhibitor to ATGL, the first step in lipolysis. And here's a still impressive reduction with the inhibitor of monoacyl glycerol lipase and hormone-sensitive lipase. So FAFAs are released from triglycerides during lipolysis, and we think this is physiologically relevant. It could explain why PASA isomers increase in white adipose tissue with fasting. Okay, so one level of regulation is storage depots for FAFAs in other lipids. What about synthesis? We're really intrigued. Are FAFAs synthesized in mammalian tissues? And to answer this, our collaborator, Alan Sagatilian, synthesized this lipid, which is we say unnatural, meaning it's not very abundant in nature. So it can be used like a tracer. And we can actually gavage mice with this lipid, and then we can analyze the serum and tissues by LCMS, looking for this lipid, the HHA, being incorporated into a FAFA. So it finds the palmitic acid, and it constitutes the hydroxy fatty acid to make a palmitic acid HHA. So here are the results I'm showing you in liver. Vehicle-treated mice don't have a significant amount of this FAFA, but the 9-HHA gavaged mice have easily detectable levels of this PAHHA. So FAFAs are synthesized in vivo. The biosynthetic pathways could provide targets to restore or increase FAFA levels in insulin-resistant people and potentially improve their insulin sensitivity. But what is the evidence that FAFAs are synthesized in people? And so Andres Kuda, I mentioned him, he drew the FAFA-ome. He carried out this study with clinical collaborators where people were given either 5 grams a day of corn oil as placebo or EPA DHA, omega-3 PUFAs, daily for 24 weeks. And then they measured serum FAFA levels. And so you can see here in black, in the placebo-treated people, some low levels of FAFAs containing the DHA, but the people who actually were treated with FAFAs, for some reason, this is not advancing. Okay, had a marked increase in the FAFAs that contained these polyunsaturated fatty acids. So FAFAs can be synthesized in vivo in humans. Now, what are the biosynthetic enzymes? So specifically, what we're looking for is an unidentified FAFA synthase, which we think is an acyltransferase or a transacylase that catalyzes this reaction to make this ester bond. And I will tell you, there's 60 plus human enzymes that are identified in RefSeq as acyltransferases. Rucha Patel, a fellow in the lab, overexpressed 18 of these with minimal effect on FAFA biosynthesis. So we went back to the drawing boards. And this morning, one of the students asked me the question, this afternoon, I guess, what do you do when nothing's working? This is an example. It just wasn't working. How many did she have to express, you know? So we went back to the drawing board. And we went back to those adipose GLUT4 overexpressing mice, the ones with enhanced glucose tolerance, increased adipose de novo lipogenesis, increased plasma fatty acids, and the mice that we did the adipose tissue global lipidomics on, which resulted in the discovery of the FAFAs. So here's the 18-fold increase in FAFA levels in these mice. And we decided to try to find the biosynthetic enzyme in the fat cells from these mice. So the approach Rucha took was to make stromal-vascular fraction-derived adipocytes from the adipose-specific GLUT4 overexpressing mice. So the first question is FAFA biosynthesis increased in the adipose GLUT4 overexpressing adipocytes? And here are the results. So here are first the wild type mice, the adipocytes from wild type. And as Rucha adds hydroxysteric acid, you can see FAFA biosynthesis increases. Now here in light blue are the adipocytes from the adipose GLUT4 overexpressing mice. So the biosynthesis of new FAFAs is increased even in the absence of extra HSA, but as she adds HSA, it's increased at every concentration. So FAFA biosynthesis is increased in adipocytes overexpressing GLUT4, and this is cell autonomous. So our assay can detect increased FAFA biosynthesis in adipocytes from the adipose GLUT4 overexpressing mice in vitro. But when we overexpress acyl transferases in the cells, we saw minimal increase in FAFA biosynthesis. So we reasoned maybe that is because there's simultaneous FAFA hydrolysis. So we had identified some FAFA hydrolases. And the ones we identified are serine proteases, and they're inhibited by this fluorophosphonate inhibitor called MAFP, which forms a covalent bond with enzymes containing a serine threonine in the catalytic domain, and this inhibits activity. So the notion was maybe we'll see increased FAFA biosynthesis if we block FAFA hydrolysis with MAFP. So here's the experiment. Again, Rucha sees increased FAFA biosynthesis just adding this substrate, hydroxysteric acid. Now, what happens when she adds that MAFP? The opposite of what we expected. The FAFA biosynthesis goes down, not up. We thought we would inhibit hydrolysis and increase FAFA biosynthesis, but it decreased it. So we thought MAFP may be blocking a biosynthetic enzyme. So we used activity-based proteomics with Alan Sagatilian, and we pulled down proteins that interact with the active site of MAFP. So this assay works like this. Rucha treats the adipocytes with the MAFP inhibitor. Here's the structure of it. Dio Siegel made this molecule for us. Then she makes cell lysates in which the proteins are bound to the inhibitor. Then she performs click chemistry to fix the label on the proteins. We send the proteins, to Alan, who then uses streptavidin to enrich and does proteomic identification of candidate enzymes. And then we developed a strategy to prioritize the proteins bound to the inhibitor. So we were looking for proteins that were more abundant in the adipose GLUT4 overexpressing adipocytes compared to wild-type adipocytes, ones that contain this serine nucleophile. And then we used gene ontology to identify proteins related to lipid biology or biochemistry. Then we use specific inhibitors of the enzymes to narrow down which ones were really affecting FAFA biosynthesis. And what emerged was adipose triglyceride lipase, also known as patatin-like phospholipase domain containing protein 2, or PNPLA2. And actually, this crystal structure in the literature is the patatin domain bound to MAFP, that tool that we used. So this molecule, MAFP, bound in the active site of ATGL, inhibits FAFA biosynthesis. So what is this family? I mean, you've probably heard a lot of PNPLA3 mutations in this gene cause metabolic dysfunction associated with steatotic liver disease. We're talking about PNPLA2, which is ATGL, which catalyzes the first step in lipolysis. And for interest of time, I'm going to not read all of this, but say that there are mutations which cause various disease conditions associated with neutral lipid storage. Many of these people with ATGL mutations have cardiomyopathy and skeletal myopathy. So is ATGL actually increased in the adipose tissue of the adipose GLUT4 overexpressing mice? Yes, it is. This is a Western blot of ATGL in wild type in HE4OX mice. So does ATGL increase FAFA biosynthesis in cells? And to carry this out, we transfected HEC cells with catalytically dead and wild-type ATGL. With the catalytically dead, there was no increase in FAFA biosynthesis, but with active ATGL, there was an increase in FAFA biosynthesis. So what if we knock out or inhibit ATGL? Does that affect FAFA biosynthesis? So here we're looking at the flox control mice, and then adipose ATGL knockout mice. So you can see they have marked reduction in FAFA biosynthesis. But we can also inhibit the biosynthesis in the floxed mouse using an inhibitor called aglastatin, and that decreases FAFA biosynthesis. And I just wanted to show you here, it's not just PASAs, it's other subfamilies of FAFAs that are also, the biosynthesis is inhibited when ATGL is knocked out or inhibited. So can ATGL synthesize FAFAs in vitro from triglyceride as an acyl donor and hydroxy fatty acid? So there's nothing else in this test tube except this triglyceride, trialine, and this acyl acceptor, 9-hydroxysteric acid, and the enzyme ATGL and its cofactor. And you can see, yes, it synthesizes this. This is oleic acid hydroxysteric. And when we use the ATGL inhibitor, it's markedly inhibited. So this enzyme can really do this FAFA biosynthesis. There's nothing else around to do it. So can we measure PASA biosynthetic activity in vivo? And if so, is it reduced in adipose ATGL knockout mice? So Rucha did this in vivo experiment. She gave mice deuterated HSA, and then she took out the tissues and she measured labeled PASA production. And you can see it's quite robust in the fat of the control flox mice, hardly see it at all in the adipose ATGL knockout mice. And at this time point, we didn't see it in liver or serum. Also, remember I said that FAFAs are incorporated into triglycerides as a storage form. That's also undetectable in adipose ATGL knockout mice. The FAFAs are not being made to be incorporated into triglycerides. So what is the physiologic relevance of this? Where if we measure steady state levels of FAFAs endogenously in the fat of the adipose-specific ATGL knockout mice, they're quite low. Here's sub-Q white fat, perigonadal white fat, brown fat, very low, not as low in the liver, but they are reduced in the serum, which makes us think that adipose tissue is a major source of circulating FAFAs. So low PASA levels in adipose tissue of adipose-specific ATGL knockout mice indicates that ATGL is an important regulator of PASA levels in vivo. So my last data, you always have to make sure it happens in people. This is human adipocytes now. We give the HSA, we stimulate FAFA biosynthesis, we then use the inhibitor, ATGL, and we at least partially inhibit that biosynthesis. So ATGL is also important in human adipocytes for FAFA biosynthesis. FAFA biosynthesis in human adipocytes is also regulated by ATGL. So these studies have shown that using functional proteomics, we identified adipose triglyceride lipase as a potential enzyme involved in FAFA biosynthesis. ATGL overexpression increases FAFA biosynthesis. ATGL inhibition or knockout reduces it. ATGL transacylase activity results in synthesis of FAFAs using a fatty acid chain of a triglyceride or a diglyceride, and levels of free FAFAs and FAFA triglycerides are low in adipose tissue from adipose ATGL knockout mice, underscoring the physiologic importance of ATGL in regulating FAFA levels. So if we look at this as a cartoon, we know that ATGL has lipase activity, but now we found that in the presence of hydroxy fatty acids, it has a completely other enzymatic activity, transacylase activity, and that activity then forms a FAFA. And I didn't show you the data, but if we drive the amount of this substrate by altering DGAT, that will also increase this FAFA biosynthesis. So this transacelase activity is a new activity for this important enzyme. And we were really interested to see that there's another family member of ATGL. Remember, ATGL is PNPLA2, but PNPLA1, which is in the skin and has a crucial role in skin barrier function, it does it by directing this acyl ceramide biosynthesis, which is also transacylation. So triglyceride is the acyl donor, different acyl acceptor for this, and here's the product. So this paper proves that this family of enzymes has physiologically important transacylase activity. So FAFAs are regulated by storage in other lipid classes, also by synthesis. ATGL is the only synthetic enzyme we've found so far. We have found hydrolases. We can talk about that in the reception if you're interested. And finally, what I've showed you is that GLUT4 in white fat is critical in terms of driving lipogenesis via ChREBP, which results in the production of FAFAs, which work through GPCRs to increase glucose transport in fat, decrease inflammation, which is important in both type 1 and type 2 diabetes, increase GLP-1 secretion, through GPR40 directly increase insulin secretion. FAFAs are in many other tissues, which also may contribute. And our more recent data shows that FAFAs decrease lipolysis, which is part of the mechanism for the decreased hepatic glucose production, and that they have direct protective effects, anti-senescence effects in pancreatic islets. So thank you for your attention. I think I mentioned these wonderful people in the lab, and some of these people have recently moved on to their independent labs. So I am recruiting new postdocs if you're interested. And you took away my ability to advance.
You want to share your slides?
This is us last summer. And these are the team in Alan Sagatilian's lab, and Dio Siegel, I mentioned, is at UCSD, a synthetic chemist who makes the lipids. I think I mentioned all of the collaborators as I went along. I do want to thank the islet donors and their families for the human islet studies. Thank you for your attention.
So we are a little bit over time. Why don't we entertain 3 questions, then we can continue the discussion at the reception. Go ahead.
Hi, I just have a question. If BASA has any effect on pro-apoptotic or anti-apoptotic in the mitochondria, as it can affect insulin secretion, and also BASA protects pancreatic beta cells?
Right. So there are effects on apoptosis. Now, if you're asking, specifically mitochondrial effects, we have not studied that, but there is one paper showing that they are anti-apoptotic.
Okay, thank you.
Go ahead, please. Yeah.
That was fantastic. Yeah. One of the things that's really impressive is, that you can remain this productive and creative throughout your career, and it's really remarkable. I had one comment and then a question related. The comment is about the hepatic glucose production. And there are a number of people, Jerry Schulman, Maury Birnbaum, for example, who think that the main effect of insulin in clams on hepatic glucose production is through suppressing lipolysis in fat. So you wonder whether the effects you're seeing of the FAFA is not directly on the liver, but by improving the health and the responsiveness of fat, and perhaps through limiting inflammation in fat to help maintain healthy fat.
So I actually have that whole paradigm on a slide. And we did show in our 2019 JCI paper, I couldn't show all the data here, that in vivo, the FAFAs reduce fatty acids
Yeah.
during the clamp more
Yeah.
than in the non-FAFA treated
Yeah.
animals. And if you prevent that by infusing intralipid, so the FAFAs, the
Yeah.
fatty acids cannot
So-
go down, you lose the
Yeah,
positive.
so another way of putting that is that they are restoring insulin sensitivity, the ability of insulin to suppress lipolysis
Right.
and the flux of fatty acids.
Yeah.
So related question, ATGL is important everywhere. You have a lipid droplet, including the liver, where, we've been studying its role in gluconeogenesis, and Jerry Shulman has shown it's important in mediating glucagon effects on gluconeogenesis. So, I wonder whether FAFA, usually it's been thought of as regulating lipolysis, fatty acid oxidation, but you think FAFAs are also being produced in the liver, and do they have local effects?
Well, I think it's very possible there are. It's interesting. So the PASA family that we've studied, there are 8 isomers in fat and only 3 isomers in the liver. But, there are now dozens of different families. So there may be other FAFAs in the liver that are important. So, yeah. Yeah.
Yeah, Dr. Kahn, thank you so much for this wonderful talk and a very exciting data. So you mentioned like these FAFAs, there are different types, different isomers in different tissues. So I'm just wondering, are there particular isomer, particular type in certain tissues are very highly correlated with this insulin sensitization, so can be used as a potential biomarker?
Okay, so the highest levels and the most isomers are in fat. And the biomarker question, I was mentioning to someone else today, we have a paper we're about to submit that a change in PASA levels will predict worsening glucose tolerance and development of impaired glucose tolerance in people, in family members of people with type 2 diabetes. So they can be used as a biomarker, but to compare and contrast, we looked at another family, we call them the PAHOA, the palmitic acid hydroxy oleic acid, and those, the initial levels predict who's going to get impaired glucose tolerance. So I think they can be used as biomarkers. Yeah.
So terrific. Thank you very much, Dr. Kahn. What can I say? The tour de force, phenomenal presentation. You made it all sound so seamless in spite of the complexity. I cannot thank her enough.